A man vanished into Lake Tahoe wearing scuba gear, and for 17 years, he simply ceased to exist. No body, no burial, no answer. Then, in the summer of 2011, a diver working the lake's deep blackwater spotted something wedged into a rock crevice 265 feet down. It was him, still in his wetsuit, still recognizable, barely touched by time. And the question that question raised was not just where he had been all those years. It was how a human body could sink into freezing darkness for nearly 2 decades and come back looking like it had been down there for months. The answer to that question would pull back the curtain on one of the strangest natural laboratories on Earth. At a place where cold water performs a kind of biology-defying trick that most people assume only happens in science fiction. And it would also open the door to something far more powerful, the science that finally lets us put names to the nameless drowned eye. The forensic genetics that turns a preserved stranger into a son, a husband, a daughter, someone's missing person, finally coming home. Lake Tahoe sits high in the Sierra Nevada, straddling California and Nevada, and it is deceptive. From the surface, it looks like postcard water, blue and clear enough that swimmers can see their own feet at absurd depths. But drop below a few hundred feet, and the character of the lake changes completely. The United States Geological Survey lists its deepest point at 1,645 feet. Making it the second deepest lake in the country. Stack the Empire State Building inside it, and water would still cover the tip. At those depths, sunlight essentially disappears, the water temperature drops to somewhere near freezing, and oxygen becomes scarce. It is, in almost every practical sense, a refrigerator the size of a small sea, and refrigeration is exactly the process at the center of this mystery. To understand why a body can last so long down there, you have to understand what destroys a body everywhere else. Decomposition is not some vague, mystical unraveling. It is biology, and specifically it is bacteria. Both the bacteria already living inside every human gut, and the bacteria that colonize a body from the environment after death. These microorganisms break down tissue by metabolizing it, and like all living things, their metabolic machinery runs on enzymes, and enzymes are temperature-dependent proteins that fold into precise molecular shapes to do their work. Drop the temperature far enough, and those enzymes slow down dramatically, some nearly stopping altogether. Add in an oxygen-poor environment, which suppresses the aerobic bacteria that do much of the fastest tissue breakdown. And you get conditions that mimic something closer to a walk-in freezer than an open grave. Lake Tahoe's cold, low-oxygen depths do precisely this. Investigators examining the 2011 case found the water at 265 feet was under 40 degrees Fahrenheit, cold enough to functionally arrest much of the decomposition process for years at a stretch. This is not unique to Tahoe. Lake Superior has its own dark reputation as a lake that never gives up her dead, and researchers have documented similar preservation phenomena in other deep, cold, low-oxygen lakes across North America. But Tahoe's story became something bigger than science. It became legend. For decades, a persistent rumor circulated that the legendary oceanographer Jacques Cousteau had dived into Tahoe's depths sometime in the 1960s or 1970s and surfaced shaken, refusing to describe what he'd seen except to say the world was not ready for it. The story spread that the lake floor was littered with hundreds, even thousands of perfectly preserved corpses, drowned gamblers, mafia hits from Nevada's casino era, maybe even the remains of Chinese railroad workers killed during the construction era of the American West. It is a haunting image, and haunting images travel fast, especially once the internet gets hold of them. But here is where the story takes its first real turn, because the evidence tells a very, different tale than the legend. Charles Goldman, a UC Davis limnologist, someone who spent his career scientifically studying the physics, chemistry, and biology of inland lakes, personally knew Cousteau and helped build the modern scientific understanding of Tahoe's ecosystem, starting in the 1960s. According to people who worked alongside him, there is no credible account that Cousteau ever made the shocking discovery attributed to him, and if he had documented mass graves at the bottom of a major American lake, it strains belief that he would have kept it quiet rather than turning it into exactly the kind of sensational, funding-generating documentary he was known for making. In 2016, an actual organized search combining human divers and remotely operated underwater vehicles, conducted by the Undersea Voyager project, scoured sections of the lake specifically looking for these rumored remains. They found none. In 2022, divers working a separate cleanup effort focused on removing trash from the lakebed also reported finding no bodies. Search and rescue professionals who have spent careers looking for drowning victims in Tahoe describe the search process itself as something closer to finding a needle in a haystack, precisely because the lake is so vast, so deep, and so dark that recovery, even when a location is roughly known, can take enormous time and specialized equipment. So if there is no field of preserved mafia victims carpeting the lakebed, what actually happened in 2011, and why does the story still matter so much? The diver recovered that year was a Reno man who had gone missing while scuba diving 17 years earlier. He was found lodged in a rock crevice at 265 feet, discovered using a small, self-powered, remotely operated vehicle equipped with a light and a camera, the same basic technology used across modern underwater forensic recovery. It took investigators the better part of a day to locate him and bring him to the surface using a mechanical claw mounted to the ROV. His own scuba gear had inadvertently helped protect him further, shielding soft tissue from the kind of scavenging that larger aquatic organisms might otherwise have carried out. Though experts are careful to note that this does not mean every body in the lake would fare the same way. Preservation at this level is possible, even fairly reliable given Tahoe's specific conditions, but it is not guaranteed and it is not evidence that dozens of unrecovered bodies remain in similarly pristine states scattered across the lakebed. This single, real, well-documented case became the seed from which the entire modern mythology grew, and that pattern is worth sitting with for a moment, because it says something important about how humans process uncertainty. A genuinely unsettling scientific fact: cold water can preserve a human body far longer than most people intuitively expect. Got picked up, exaggerated, and reshaped into an urban legend involving monsters, mobsters, and a world-famous explorer who almost certainly never said what he is quoted as saying. Reality was strange enough on its own. It did not need embellishment, but the embellishment happened anyway, and once it did, separating fact from folklore required exactly the kind of rigorous, evidence-based thinking that defines forensic science itself. Think about why that particular embellishment took hold so easily. Human beings are pattern-seeking creatures, wired to fill uncertainty with narrative, and a deep, dark lake with no visible bottom is close to a perfect canvas for that instinct. We cannot see down there. We cannot verify or disprove a claim about what lies in that darkness without enormous technical effort, expensive equipment, and trained divers willing to risk nitrogen narcosis and crushing pressure. And even then, a single expedition can only cover a tiny fraction of a lakebed spanning tens of square miles. That gap between what is knowable in principle and what has actually been observed in practice is exactly the space where legends thrive. It is the same gap that once let sailors believe in sea serpents charted on the edges of unexplored maps, and it is the same gap that, closer to home, lets one verified case of a preserved diver metastasize into a folk belief that an entire lake floor is a mass grave. Science does not eliminate mystery by refusing to engage with it. It eliminates mystery by sending the remotely operated vehicle down, by measuring the actual water temperature at depth, by testing the real chemistry of decomposition rather than accepting the most dramatic available story. And this is where the story stops being about a lake and starts being about the extraordinary tools modern science has developed to answer the question that lurks underneath every one of these tales. Who was this person? Because recovering a preserved body is only the beginning. A face altered by years underwater, clothing degraded, personal effects lost or unrecognizable, none. Of that reliably tells you a name. What tells you a name with a level of certainty that would have been unimaginable a century ago is DNA. Every cell in the human body carries roughly 3 billion base pairs of genetic code, wound into 23 pairs of chromosomes, spelled out in only 4 chemical letters: adenine, thymine, guanine, and cytosine. Pairing together in a double helix, first described by James Watson and Francis Crick in 1953, built on X-ray diffraction data captured by Rosalind Franklin, whose photographs reveal the helical structure before her contribution was properly credited by history. That structure is not just a beautiful molecule. It is an identification system more precise than a fingerprint, more permanent than a face, and remarkably durable, even under conditions that would destroy almost everything else about a person. Forensic geneticists exploit specific regions of the genome called short tandem repeats, small sequences of DNA that repeat a variable number of times from person to person. The FBI's forensic DNA database in the United States relies on a standardized set of these repeat regions precisely because they vary enormously between unrelated individuals while remaining stable enough within one person's own cells to serve as a reliable signature. Compare the repeat pattern from recovered remains against a reference sample, whether that is a toothbrush from a missing person's home, a blood relative's cheek swab, or an old evidence sample from a decades-old case file. And investigators can calculate a match probability so precise that courts around the world now accept it as scientific fact rather than statistical suggestion. It is worth pausing on how strange this actually is, biologically speaking. Every one of the trillions of cells in your body, from a strand of hair to a flake of skin to the marrow inside a femur, carries the same complete genome, the same 3 billion letter instruction manual, faithfully copied every single time a cell divides through a process. Called DNA replication, in which the double helix unzips down the middle and an enzyme called DNA polymerase builds a fresh complementary strand along each half, checking its own work for errors as it goes. That astonishing internal proofreading system is why your genetic signature at 19 years old is, for identification purposes, functionally the same signature as at 60. And it is why a genetic profile pulled from a fragment of bone recovered from icy water can still be compared, decades later, against a cheek swab taken from a living relative who was not even born when the person went missing. The code does not fade the way a photograph fades or a memory blurs. It either survives chemically intact enough to be read, or it does not. There is very little in between, and that binary nature is precisely what makes DNA identification so much more reliable than dental records, personal effects, or eyewitness recollection, all of which degrade gradually and can mislead investigators with false confidence. But cold water does something else to remains beyond simply slowing bacterial decay, and it matters enormously for genetic identification. DNA itself degrades over time through a process driven largely by hydrolysis and oxidative damage, chemical reactions that break the bonds holding the genetic code together. And like the enzymatic reactions that drive decomposition, these degradation reactions are also temperature sensitive. Cold, oxygen poor, low light environments slow this molecular unraveling dramatically. This is part of why scientists have successfully extracted genetic material from remains recovered in cold environments that are decades, sometimes centuries old. And it connects directly to one of the most breathtaking frontiers in modern genetics, ancient DNA research, where scientists have sequenced genomes from remains found frozen in permafrost and preserved in cold cave systems for tens of thousands of years, remains belonging not just to ancient humans but to our extinct evolutionary cousins, the Neanderthals, whose genetic legacy still lives on today, quietly present in the genome of nearly every person of non-African descent walking the planet right now. The same physics that let a drowning victim remain recognizable in Tahoe after 17 years is, at a much larger scale and over a much longer timeline, the same physics that has let paleogeneticists reconstruct the genome of a Neanderthal woman from a 40,000-year-old bone fragment. Cold is not just a preservative for tissue, it is a preservative for information, and DNA is, at its core, information, an instruction manual for building and maintaining a living thing, written in a chemical code that nature has been running continuously for roughly 3.5 billion years. This is precisely why forensic identification of remains recovered from cold water environments carries a particular kind of hope for families of the missing. When a body surfaces from Tahoe's depths after years or even decades, the recognizable clothing and remaining features may already offer investigators a strong initial hypothesis, but it is genetic testing that converts hypothesis into certainty. Forensic laboratories extract DNA from available tissue, sometimes from bone marrow, sometimes from teeth, which are remarkably resistant to degradation because the hard mineral structure of dentin and enamel protects the genetic material inside far longer than soft tissue can protect it. That extracted profile is then compared against databases like CODIS in the United States, the Combined DNA Index System, which links local, state, and national forensic databases together, or matched directly against DNA voluntarily submitted by family members of a specific missing person. When the numbers align, when the statistical probability of a random match becomes vanishingly small, families who have spent years, sometimes decades, in a state of unresolved grief finally receive something that no rumor about mobsters or lake monsters could ever offer them: an answer. This is the quieter, more human side of the Tahoe story, one that rarely gets the same viral attention as the ghost stories. Every unresolved missing persons case involving open water is, statistically, a case where a body may exist in conditions capable of unusually long preservation, and every advance in forensic genetic technology increases the odds that a family finally gets closure. Genetic genealogy techniques, the same category of tools that solved cold cases involving the Golden State Killer by tracing distant genetic relatives through public ancestry databases, have increasingly been applied not just to catching perpetrators, but to identifying unnamed remains recovered from exactly these kinds of extreme environments, whether that is a lake, a mountainside, or a shipwreck. Investigators upload a genetic profile from unidentified remains into genealogy databases, search for distant relatives, sharing small segments of matching DNA, and painstakingly build family trees backward until they can triangulate a name. It is genetics as detective work, using the mathematics of shared ancestry, the same mathematics that explains why you share roughly 50% of your DNA with a parent, 25% with a grandparent, and smaller, more diffuse fractions with cousins, several generations removed, to solve mysteries that once seemed permanently unsolvable. And this raises a genuinely difficult question that the scientific and legal communities are still actively working through. If a stranger's Preserved remains can be identified through distant relatives who never consented to have their own DNA used this way, because a second cousin uploaded a saliva sample to a consumer ancestry service purely out of curiosity about where their family came from. What are the actual boundaries of genetic privacy in an era where identification, no longer requires your own direct participation? Researchers, ethicists, and law enforcement agencies continue to debate where the line should sit between the undeniable public good of identifying human remains and reuniting them with grieving families, and the privacy expectations of people who submitted their own DNA for entirely unrelated personal reasons, like tracing immigrant ancestry or checking for inherited disease risk. There is no settled global consensus on this yet. Some countries restrict law enforcement access to genetic genealogy databases far more tightly than the United States does, and several consumer ancestry companies have changed their own policies over the years in response to public backlash, some opting into law enforcement cooperation only with explicit customer consent, others resisting such requests entirely unless compelled by a court order. It remains one of the most consequential open ethical questions in modern forensic science, precisely because the technology has advanced faster than the legal and philosophical frameworks meant to govern it, and researchers, bioethicists, and defense attorneys continue to debate where exactly the boundary should sit between the compelling public good of identifying the dead and reuniting them with grieving families, and the reasonable expectation that submitting your own DNA for a hobbyist curiosity about your ancestry should not silently make your third cousin identifiable to a government agency without their knowledge or consent. There is also a quieter, more personal dimension to this debate that rarely makes headlines. Every person who has ever submitted a saliva sample to trace their family history, expecting nothing more than a breakdown of regional ancestry percentages or a handful of distant relative matches, has effectively become a permanent, unwitting participant in a nationwide identification network they never explicitly signed up to join. That is not necessarily a bad thing. It has already helped resolve missing persons cases that had gone cold for decades, giving families answers that no amount of hoping or searching ever could. But it does mean that the concept of genetic privacy, once understood as something an individual controlled entirely on their own, has become something closer to a shared, networked property of an entire extended family, stretching across generations and continents, whether every member of that family is aware of it or not. Step back from the ethics for a moment and consider just how extraordinary it is that any of this is possible at all. A century ago, a body recovered from the bottom of a mountain lake with no identification on it would very likely have remained forever unnamed, filed away as a Jane or John Doe, a tragedy without resolution. The scientific journey that changed this did not happen overnight. It began with Gregor Mendel, a 19th century Augustinian friar, quietly breeding pea plants in a monastery garden, meticulously tracking how traits like flower color and seed shape passed from one generation to the next, laying down the mathematical laws of inheritance decades before anyone understood what a gene physically was. It continued through the 20th century as scientists slowly identified DNA rather than protein as the actual molecule of heredity. Then Watson, Crick, and Franklin's revelation of its double helix structure. Then decades of painstaking work learning to read, copy, and eventually sequence that code. It reached a historic milestone with the Human Genome Project, an international scientific collaboration completed in the early 2000s that mapped essentially the entire sequence of human DNA, a feat that took over a decade and billions of dollars, and today can be functionally approximated for a single genome in a matter of days for a fraction of the original cost. Every piece of forensic genetic identification happening today, whether in a Tahoe recovery case or a 100-year-old unidentified skeleton, stands on top of this staggering accumulated history of scientific labor. It is easy to forget just how recently all of this became possible. Barbara McClintock spent decades of painstaking and often dismissed research on the genetics of maize before the scientific establishment finally recognized that genes themselves could move within a genome, jumping from one location to another, a discovery so far ahead of its time that she did not receive the Nobel Prize for it until 1983, more than 3 decades after her key findings. Her work quietly reshaped how scientists understood the genome not as a fixed, static blueprint, but as something dynamic, capable of rearrangement, a concept that underlies much of how modern researchers now think about mutation. Genetic disorders, and even cancer genetics, where cells accumulate genetic changes over a lifetime until normal growth control breaks down entirely. And decades after McClintock, in the early 2010s, biochemists Jennifer Doudna and